First of all, I'd like to thank the organizer for inviting me to talk about aberrant inflammation in chronic HIV infection today. With the life expectancy of people living with HIV that is improving a lot, especially over the past 25 years, there is an increase development of age-related disease and aging in those populations. So even if the life expectancy has improved, there is still a gap. And this gap in high-income countries is between 4 and 10 years, and it depends on CD4 cells levels, as shown on the graph here, gender, time of art initiation, and this age-related disease that are increasing in people living with HIV are related to immune activation, as the previous speaker explained to you, and these many comorbidities are related to non-communicable disease like cancer, cardiovascular disease, hypertension, also communicable disease like other viral infection like hepatitis, cytomegalovirus, and also mental health disorder. This immune activation that persists has been shown to be there even if the people control the virus, as shown in red and green here, in red for HIV controllers and in green for people under ART treatment. The immune activation still persists. It also persists with residual chronic inflammation, as shown on the graph here. When you start the ART treatment in the gray box, the viral load decrease in black. The CD4-CD8 ratio, which is a marker of progression, increase but don't return to the level that you had before infection. The CD4 cell count partially recovered, but the activation shown here by the level of activated CD8 T cells or the inflammation doesn't fully return to baseline level. And this leads to the development of non-HIV comorbidities. I mean, the risk of developing such a disease increase. And the chronic inflammation might have multiple causes. As explained by before, the multiple causes are the first thing was the reservoir, because some virus still can be produced randomly by activated cells. Are toxicity, but it's less and less true. Cytomegalovirus and other copathogens, the loss of the immunoregulation, and also the microbial translocation through the damaged intestinal barrier. Those are the major facts that could lead to this the residual chronic inflammation that persists even under ART treatment. About the reservoir, why there is still some virus that could replicate under the treatment? Well, it has been shown that in some tissues, like here in the lymph node and the gut, the antiretroviral drug doesn't completely reach this tissue. So there is suboptimal concentration, and so the virus might sometimes still replicate. Moreover, the virus under our treatment will replicate in follicular CD4 T cells, which are cells in B cell follicle, and these cells are out of reach of cytotoxic CD8 T cell to kill them. The other inducer of the immune inflammation could be the soluble GP120. Some infected cell and even the variant could produce soluble GP120, and this GP120 could be detected in the plasma and tissue from people living with HIV, even with the ART treatment, where we still detect high level of GP120 in the spleen and in the lymph node. GP120 binds to CD4 molecule on monocyte, macrophage, T cell, dendritic cell, and this would lead to the production of a panel of cytokine, and this leads to the sustained immune dysfunction and inflammation. So how to control this residual immune inflammation that persists? Well, one of the strategies that has been used is to treat very early. So early initiation of antiretroviral treatment favors first the reduction of the viral reservoir and the viral diversity. It allows also to lower the inflammation level, and early ART avoids also most tissue damage, but not all. So even if we start, it has been shown that even if you start the ART early, there is still some mucosal damage persisting and residual also chronic inflammation. So there will still be some increase of some comorbidities. Other strategy to treat immune activation and inflammation have been tested, and here are a panel of examples, but it's not exhaustive, such as treatment that target the microbial translocation with probiotic and prebiotic, for instance, to maintain gut homeostasis. Some immunosuppressive drugs like prednisolone, cyclosporin A, hypoglycemic agents, statins like pitavastatin, which give good results in a RETRIEVE trial to reduce cardiovascular disease, and some other treatment like temsavir that target GP120 itself. In the lab where I work, we like to learn from nature, and we learn a lot of important clues from the natural host of HIV, in fact, the African green monkey. Why? Because in this natural host, which it's not the only one, you know, it's a natural host, and mostly a monkey from Africa. There is more than 45 species which are infected and do not develop AIDS. So we are working on African green monkey and compare it to the macaque model who develop AIDS when infected with HIV. And what we see in the African green monkeys, they have high viremia, like in the pathogenic model. They lose their CD4 T cells during the acute phase of infection, and they develop an acute inflammation, but when you look at the chronic phase, they don't have inflammation. So they find a way to control the inflammation during the chronic phase of infection. And moreover, in the natural host, it has been shown there is no damage of the intestinal barrier, no microbial translocation, and the lymph nodes are preserved. In the lab, indeed, we studied the lymph node of the natural host and compared it to the pathogenic infection. Here, if you look at the lymph node of macaques in chronic HIV infection, what you see on the left picture, it's in red. The HIV is all over the B cell follicle, and it leads to this persistent presence of the SIV in the B cell follicle and gender hyperplastic B cell follicles. And we have also persistent high level of inflammation that leads to collagen deposition and fibrosis. You have to know that the lymph nodes are a training center for the immune system, and being always activated is not good because you will have the chronic inflammation because of this. And when looking at the natural host during the chronic SIV infection, what you see, it's clear on the picture on the left in the bottom, you don't have SIV or very few SIV in the B cell follicle. And when we look at the NK cells, which are killers of the innate immune system, they kill infected cells. So here, when you look at NK cells, you see them, they are there. They are in the B cell follicles. So it seems like the natural host, I found a way to control the training center of the immune system, to not have SIV, to not have too much inflammation in the center, and so you don't have fibrosis and chronic inflammation. So nowadays, there is a study trying to induce these NK cells to have something similar to what we see in natural host, like in this study from Mikko Pagliardini, where they use IL-21 and interferon to induce this NK cell, to activate these NK cells. And what they show, it's in blue in the figure, it's this treatment in macaque that had the heart, induce indeed mature NK cells and activation of NK cell, leading to a decrease of the replication competent virus in the lymph node, which is beneficial. So a lot of studies today aim to improve NK cell function, like with IL-15, TLR agonist, and so on. Most of those strategies are based on the oncology research. I will take the last 2 minutes to talk about our last development, our last study with the natural host. By studying the natural host, we saw also that we identify some special cells in the gut of the natural host. It's CD8 T cells with expressing NKG2A. And we saw them induced in the gut of natural host as compared to the pathogenic model where we don't see them. As I told you before, natural host, even if they lose partially their CD4 T cell in the intestine, they don't have a disruption of the intestinal barrier, and there is no microbial translocation. So by using the glatiramer acetate, so glatiramer acetate is a molecule that has been used for many years in multiple sclerosis. It's an immunomodulator that has been shown to induce this type of CD8 T cells. It also has been shown to reduce expression of pro-inflammatory cytokine in inflammatory bowel disease, for instance, to modulate CD4 T cell response and to activate NK cell lysis. So we thought it might be a good candidate in our study. So we conduct a large preclinical study with glatiramer acetate with different group of macaques that we treat with this molecule during the acute phase, the chronic phase, and combining with antiretroviral treatment. And to make a long story short, what we found is that comparing to the controls, indeed, the treatment with glatiramer acetate was beneficial. We observe an increase of CD4 T cells in almost all tissue, and the CD4-CD8 ratio, and a decrease of inflammation showed on the right of the slide. So finally, I would like to thank all the people working in the lab and all our collaborators. Thank you.